that's essentially what John Gurdon did in the 50s, 60s with xenopus embryos. The other, which Yamanaka promoted, was taking a somatic cell, again, has a nucleus in which mutations have occurred, and using these various transcription factors, OCT4, KLF4, SOX2, CMEC, and now there are variations on this thing. One with a certain frequency can transform or convert somatic cells back to an earlier progenitive stage or an embryonic stem cell stage, and then use that for therapeutic purposes. As I said, they're inherent problems, and the first example of somatic cell nuclear transfer in mammals was Dolly the sheep. Those of you who were premature, died young, and she had, arthritis in wrong limb and joints in which sheep normally don't get arthritis in. She was obese. She was very sick and had to be put away at about half the normal lifespan of a sheep. So there were problems. What I will point out, and which is interesting, is that her progeny turned out to be perfectly normal, had normal lives, and were fertile. So that's a caveat there. So one of the issues relating to this type of regeneration, shortness, it triggers senescence, cell senescence. And this is important in the whole aging process. Turns out that's reversible, and you see that reversible in some cancers. Same with premature senescence, which is a consequence of short telomeres. There are epigenetic changes that can be reprogrammed when a somatic cell is put into an egg environment or is induced to undergo IPS type of transformation. But it's interesting, even though you can generate embryonic stem cells from such, from fibroblasts or keratinocytes or ovarian cells, many epigenetic marks are removed, but they retain a signature indicative of the lineage that they arose from. But after they go through the germline, next generation, those seem to be removed. Imprinting is an important issue, and what I'm going to talk about is loss of heterozygosity as a consequence of mitotic recombination and that kind of mutation, which is not reversible. And one take-home message I'll tell you now, since I've made that introduction, is that if you have loss of heterozygosity, that includes, and recombination that includes, let's say, tumor suppressor, then if you're using cells that are iPS-derived or nuclear transfer derived, and they've undergone loss of heterozygosity containing a tumor suppressor, those cells are at much higher risk now for developing cancer as donor cells. So that's something that needs to be kept in mind. So how do somatic cells and embryonic stem cells differ? Salvage enzyme and purine biosynthesis, purine nucleotide biosynthesis. But what we find is that when animals, and we use a mouse model, a knockout mouse model, when analyzed in the heterozygous state, mutant frequency is about 10 to the minus 4, 1 in 10,000. That's very, very high. 80% of the events are due to mitotic recombination or recombination between chromosome homologs. Now, this is sort of a thought process. If there are heterozygous loci between the reporter gene, which in this case is APRT, and the site at which crossing over recombination has occurred, there are multiple affected loci. There are multiple loci that are heterozygous between the crossover point and the end of the chromosome. So if you do the math, there are about 20 autosomes, 19 in mouse, 22 in humans. If this happens on all chromosomes, and assume there are 10 loci merged conservatively that are heterozygous, that will be affected because they occur those loci between the crossover point and the reporter. Then you've got this number, 10 to the minus 4 frequency times 19 in mouse times 10 such loci. It gives you a mutant frequency, a predicted mutant frequency of 1 in 50, which, as I said, is unacceptable in a germline by far, and it's hard to accept in somatic cells. But I'll show you the data that we have in mouse, and I'll tell you that we've done the same in a human population, and it bears out the frequency and the type of mutation. So it's in a 129 strain background, and both alleles have been knocked out. And we cross that with a mouse of a different strain, C3H, that's wild type for APRT. So the outcome, the F1, is going to be heterozygous at APRT. Sorry. It's going to be heterozygous at APRT. We have used different strains on purpose because there are polymorphic markers that we can detect that flank the gene that we're using as a reporter, that flank APRT. So we do that, as I said, intentionally. So they'll be heterozygous along the whole length of the chromosome for these polymorphic markers. And these serve as markers for analysis. Okay, so we take skin fibroblasts or T cells and put them into culture. And we use these two cells because those are the only two cells that we've been able, at that time, were able to propagate enough to do the analysis. There wasn't any intellectual reason for doing it other than practical. But what we do with these is we put them in culture and immediately select for colonies that are APRT deficient. So they start out as APRT heterozygotes in the mouse, and what we're doing is we're taking events that have occurred in the mouse and expanding those events in culture. And we pick these colonies, we quantify them, and then we pick them and we analyze the mechanism by which they've come about. Okay, this is a stick diagram that describes how that works. So you have a parent that is in a C3H background and has wild type of APRT, and you've got the APRT knockout, green is the APRT gene, the knockout version, and you have these markers, these polymorphic markers, simple sequence repeats that you can use. And look at all of these various outcomes, but what we find is that in mouse, 70 to 80% of the events are the following. They've lost heterozygosity at APRT, and they've lost heterozygosity at flanking marks up to a certain point, and that point varies from colony to colony that we pick, and then there's retention of heterozygosity. So all these markers will have undergone loss of heterozygosity, have undergone loss of heterozygosity. So another way of looking at it, again in schematic form, is here is a pair of homologous chromosomes in a cell. You have replication. You get exchange, reciprocal exchange between these two this chromatid and this chromatid. You can see the reciprocal exchange. And now this chromatid segregates with this and this with that. This is the outcome. You can see there's loss of heterozygosity, including the marker of interest, and then retention of heterozygosity. So that's the premise. So let me just now show you a summary of the large number of animals that we've done. This is one representative experiment. And we looked comparing fibroblasts and T cell. So the red bars is the mutant frequency that we see, and the yellow bar is the proportion of events, the frequency with which we see loss of heterozygosity due to mitotic recombination. And as you can see in fibroblasts, about 80% of the events, of these mutagenic events, loss of APRT activity in this case, is due to recombination. T-cell mutant frequency is always a little bit lower, and you can see 70–75% of the events are due to mitotic recombination. That's not statistically significant for any one experiment, but every experiment we've done has shown the same trend. So what happens if we look at the same, ask the same question in a background that has lost mismatched repair capacity? In this case, we use the absence of MLH1.